Today we are going to make insulin through a genetically modified organism. An organism is any living thing, like me and you and a tree and flowers too. Not like a rock. All organisms are made up of cells. This is a cell you would find in an animal, like a human. In the cell there is DNA. Your DNA is what makes you, you. This is a cell of a bacteria, another type of organism. In bacteria, some of the DNA is in circles. These are called plasmids, and we can use them to insert a gene into the bacteria.
Bacteria have small loops of DNA called plasmids. In genetic engineering, plasmids can be cut open with restriction enzymes, and DNA from other organisms can be inserted. Restriction enzymes recognize specific nucleotide sequences at which they make cuts in the DNA molecule. They cut in such a way as to leave jagged ends with unpaired bases. These are called sticky ends because they will hydrogen bond to complementary nucleotides. A section of foreign DNA, with the appropriate bases on its own sticky ends, can bind to the plasmid, and DNA ligase helps join the ends. The result is recombinant DNA.
Then we can put the plasmid with the insulin gene back into the bacteria. The bacteria is now a genetically modified organism because it's a living thing and we have modified or changed its genes. The bacteria duplicates again and again to make many, many bacteria with the insulin gene. The bacteria have a human insulin gene, so they make human insulin. The scientists can take this insulin and give it to people with diabetes.
What do humans, flies, and worms have in common? More than you might think. In fact, even scientists have been amazed to find that the genes that control early development in fruit flies and humans are essentially identical. And genes associated with heart disease in humans have the same metabolic functions as related genes in yeast. As a result, model organisms have become even more important and relevant to health research.
Welcome to my lab. In the Lin Lab, we spend our time trying to understand neurodegenerative diseases like Alzheimer's, Parkinson's, and prions, also called mad cow disease. Because at the DNA level, so many similarities exist between complex organisms like mammals and very simple organisms like yeast, I can use yeast as a model organism to study human disease. Daily, I use the elegantly simple but incredibly powerful tools of genetic engineering described in the earlier parts of this video to make new plasmids containing human genes or pieces of human genes. The second step is to transform or put these plasmids into yeast to study the impact of each new gene. Later today, you'll be transforming yeast with your own plasmids. The following should provide a good primer before you get to work. During transformation, the cell wall, a cell's defense mechanism, is broken down so that large plasmids can pass through. It is essential to insert plasmids into healthy cells because this process introduces a huge amount of stress. A small amount of growing yeast strain is transferred to a large volume of media and grown overnight. In these conditions, cells undergo uninhibited, rapid growth. The life cycle of yeast is only 2 hours. This means that a yeast cell makes an exact copy of itself in a very short amount of time. This property is one of the major advantages of using yeast as a model organism. After the starter culture is grown overnight at 30 degrees Celsius with rapid shaking, the media, which now contains millions of cells, is divided into sterile plastic tubes and put in a centrifuge. Centrifugation separates the liquid media from the cell pellet without killing the cells. The pink pellet is made up of the cells that will eventually take up our plasmids. The liquid or supernatant is poured off of the pellet into a waste container. The pellet is dissolved or washed with water and transferred into a single tube before centrifuging again. After centrifugation, the supernatant is poured into waste to leave the washed pellet at the bottom of the tube. Next, the yeast cell pellet is resuspended in 1 milliliter of a buffer containing lithium acetate. Lithium acetate is a salt that begins to break down the yeast cell wall enough that plasmids will eventually be able to get through, but not enough to kill the cells. When all clumps of cells are dissolved in the buffer, the cells are transferred to a smaller tube to store on ice. The other two ingredients required for transformation are the plasmid and a solution of single-stranded DNA. 50 microliters of a solution of single-stranded DNA in water is combined with 100 microliters of yeast cells. Generally, transformations are completed with a very small amount of plasmid DNA. The large amount of single-stranded DNA is used as a carrier molecule to assist the small amount of plasmid across the cell wall and cell membrane into the cytoplasm. A single microliter of plasmid DNA is added to this mixture and vortex to mix the three components. 600 microliters of a polyethylene glycol or PEG solution is added to the yeast to further break down the cell wall and increase the chances of plasmid uptake. The cells, DNA, lithium acetate, and PEG are incubated at 30 degrees with shaking for half an hour. After incubation, 70 microliters of DMSO is added to the cell mixture. DMSO is a solvent that will dissolve and carry almost anything across the membrane, including plasmids. The mixture is incubated at 42 degrees for 15 minutes. This process is called a heat shock. Yeast cells thrive at 30 degrees and will die if kept at 42 degrees for a long time. But during the cell survival response, the cell wall becomes permeable and contains holes that are usually not there. We force the cells into stress so that the plasmid can pass through these holes. Following heat shock, the cells are placed on ice to recover. Cells are pelleted a final time and resuspended in buffer with nutrients to encourage growth. The final step in a transformation is to plate the cells on a nutrient-rich plate so that the transformed cells containing the genetically engineered plasmid can grow. Specific agar media is used that allows only yeast cells that have taken up plasmid to grow. This process is called selection. A laminar flow hood is used to plate cells because it is very easy to contaminate plates at this stage. I use sterile glass beads to spread 200 microliters of cells on the plate. Used beads are poured in a beaker and the plate is inverted and placed in a 30-degree incubator. It takes 4 to 5 days for yeast colonies to form, so next week we will begin to analyze your plates. Your plates will look something like this with different colony colors or phenotypes. You will learn how the red and white phenotypes correlate with processes observed in neurodegenerative disease.
Teachers face a great challenge. One teacher described that within a single class period, he spends 5 to 10 minutes doing a warm-up exercise, 5 minutes doing a conclusion activity, and 20 minutes managing his classroom, which leaves only 15 minutes to teach a lesson. We interviewed 3 teachers to learn how to create constructive teaching environments. Here's what we found.